Yesterday, prokaryotes are, of course, living things. And hopefully, by this point in the year, you know what a prokaryote is. It is a cell that does not contain membrane-bound organelles. That means that they don't have any nuclei, they don't have a nucleus, they don't have endoplasmic reticulum. They don't have Golgi bodies, right? The only membrane-bound thing that they have is the actual cell. The cell is contained within a membrane. They do have some organelles. They have the two non-membrane-bound organelles, which are ribosomes. We know that ribosomes are unique, right, for bacteria. And we know they've got cytoskeleton. But other than that, no other organelles in there. They keep their nucleus or their DNA in a region that's called the nucleated region that's slightly separated from some of the stuff that goes on in the cell, but for the most part, there's no separation. They're very small. They tend to be between 1 and 5 micrometers in diameter. And I know that doesn't mean anything to you because it's a small number, right? But for reference, eukaryotic cells tend to be between 10 and 200 micrometers. So that means that the largest prokaryote is roughly half the size of the smallest eukaryote, right? So they're pretty small, and because they're small, they're also very simple. When they divide through this process is called binary fission, and when you compare binary fission to mitosis, the end result is the same, meaning that the genetic material gets divided between two cells, but it's much simpler because it doesn't have to worry about moving organelles to different areas, and it doesn't have to worry about moving a whole bunch of chromosomes because there's only one chromosome and it's circular, right? So binary fission is the method of reproduction, and binary fission is simple and fast. Okay? The generational span for a bacteria is roughly, or bacteria or archaea, is on average about 30 minutes. So it takes about 30 minutes for them to divide once and then go through their entire life cycle and then divide again, right? So the average generational span is 30 minutes. Okay, what this means is that every 30 minutes, bacterial colony will double in size, right? If the conditions are good, right? So to put that into perspective, we start with 250 bacteria, right? In 30 minutes, there's 500. In an hour, there's 1,000, right? And then hour and a half, 2 hours, 2.5, 3, 3.5, 4, 4.5, 5, 5.5, 6, 6.5, 7, 7.5, 8, 8.5, 9, 9.5, 10. So in 10 hours, 250 bacteria become 262 million bacteria. That's a lot, right? So you can see here how if you were to, like, I don't know, leave food out on a table at a party or something like that, and there was a little bit of bacteria on it, that after 6 hours of sitting out, there's significantly more bacteria on it than there were at the beginning, and that's the kind of thing that can make you sick, especially if conditions are good, meaning that it's warm, it's not in the refrigerator or anything like that, and there's a food source. And generally, if it's on food, there is a food source, right? It's the food. So this is how you would get sick from eating something that's been left out for a while. Okay. Let's talk about the shapes of all these bacteria things. There's 3 prokaryotic shapes. We talked about those already, so we'll just cruise through these. There's cocci, bacilli, and spirilla. Cocci is spherical, bacilli is rod-shaped, and spirilla is spiral-shaped, right? And we talked about the advantages of these, that cocci tend to be smaller in total volume because as the volume of a sphere increases, the surface area decreases significantly as relative to the volume, right? And if you elongate it, that doesn't happen as much, and if you elongate it a whole bunch, then that's not really a problem. So spirilla can have the most interior volume without sacrificing too much of its surface area because it's significantly elongated, right? One of the main ways that we differentiate between bacteria and archaea is by looking at their cell walls. If you remember from yesterday, or from 2 days ago rather, one of the defining characteristics of bacterial cell walls that they had is that they had to contain peptidoglycan, right? And so we define bacteria as being in 2 different groups, a group that we call gram-positive and a group that we call gram-negative. So we can say bacterial cell walls always contain peptidoglycan, in some concentration. And then we differentiate based on how much peptidoglycan there is, right? So if a bacteria is gram-positive, essentially what happens is we have this purple stain that's called gram stain, and it's absorbed by peptidoglycan, right? And so if you put this gram stain on bacteria that have a lot of peptidoglycan in their cell wall, they turn purple because it's a purple stain and it's absorbed by the cell wall, and it turns the bacterial colony purple then, right? So gram stain is absorbed by peptidoglycan, turning the colony purple. So if it's purple, we say that it is gram-positive. And if it's gram-positive, that means we know that the concentration is high for peptidoglycan. We also know that the concentration of the other component of the cell wall, which is called lipopolysaccharide, is either low or 0, which will be important in just a minute. Okay. The other type of bacteria is called gram-negative. Gram-negative bacteria doesn't have as much peptidoglycan, and therefore the gram stain is not absorbed as much. And so instead of turning purple, it will turn more like light pink color. Okay, so the gram stain is not fully absorbed, turning the colony light pink. It's going to be absorbed a little bit because all bacteria have a little bit of peptidoglycan in their cell wall, but not nearly as much as it would be absorbed in the gram-positive, right? So if we get a gram-negative or a pink bacteria, we know that the concentration is low for peptidoglycan, and the concentration is high for this other constituent, which is called lipopolysaccharide. This is important because it's a really easy test to do, right? If you get a culture of bacteria, you just put it in a petri dish, let it divide for a while, a few hours, put it in an incubator with some food, let it divide, and it'll make a nice colony, and then you just wash it, you put some gram stain over the top of it. If it turns purple, you know that it's gram positive. If it turns pink, then you know that it's gram negative. And you can immediately eliminate like 50% of the types of bacteria that it could be, right? And then you look at things like, does it have a slime coat? Does it have any coloration to it prior to staining? And did it have a shape, a specific shape? And you can narrow down the type of bacteria that you're looking at pretty easily then, right? It's not as easy as like looking at a cheetah or something and being like, oh, look, it's got spots or stripes or whatever, right? And then you can tell what type of animal it is. Bacteria are all very small, and so it's difficult to differentiate between them. So we have to use tests like gram staining in order to figure that out. Okay. The archaeal cell walls are diverse. They're very diverse, but they're always made out of branched lipids. Okay. All right, diverse. Branched lipid compounds. Okay, and you probably don't need to know this, but when I say branched lipids, whenever we talk about carbon chains in organic chemistry, you refer to them as either branched or unbranched. So if it was unbranched, right, you would expect them to be like straight chains like this. Okay, but if it was branched, it's just like it sounds, like it looks something like this, where this continues on, but then there's also a branch that comes off of here, and there's a branch that maybe comes off of here, like that, and there might be another branch somewhere else. Okay, and so this is a branched lipid chain. So instead of just being a normal fatty acid tail like this, it's branched, and this results in some unique characteristics like the ability to overlap and sort of tangle together and form a more rigid structure, also higher melting points and things like that. Okay. What else? Let's talk about how these things make you sick, right? So if a prokaryote were to make you sick, we would call that prokaryote a pathogen. Pathogens are, like I said, cells or things that make you sick. Anything that makes you sick could be referred to as a pathogen. Anything that makes you sick or causes disease. When we talk about bacteria, roughly 1% of bacterial species cause disease and are pathogenic. Okay, most of them are not. 99% of bacteria are not pathogenic and won't make you sick. So let's talk about hand sanitizer for a sec. We're gonna bash on hand sanitizer a little bit today. So here's this stuff, and it says kills 99.99% of germs. Right? First of all, it kills 100% of germs. This is 99.99% thing is a legal issue where they don't want to, like, somebody to try to sue them because they misused it and it didn't kill some germ. But the very nature of alcohol kills things, right? It kills bacteria. It dissolves their plasma membranes, and so they can't really do anything about it. They'll die. Okay? But if you kill 99.9% of germs, you're also killing the rest of the bacteria, right? And we figured out that 1% of bacterial species are pathogenic, but 99% are not. So right now, my hands could be covered in bacteria, right? But it doesn't matter because it's a very good chance that those bacteria that are on there are non-pathogenic, right? In fact, there is some commensalistic, it's a symbiotic type, it's a type of symbiotic relationship, and there are some commensalistic bacteria that live on the surface of my skin that are adapted specifically for the conditions of the surface of my skin. And so they are better off than if I were to touch this and it had like staph on it or something, and I'm not going to get a staph infection because now I've got staph on my hand, but I've also got all this other bacteria on my hand, and the staph have to compete with the other bacteria for space and for food, right? And so the other bacteria, the good bacteria, the kind that don't cause sickness or disease, those ones are going to outcompete the staph 99.9% of the time, right? So do I want to be using this stuff? I don't know, right? Now, I gotta say, like, as you go around the school, there's a lot of disease-causing bacteria all over the place, like all of the doors and handles and everything that you basically touch, your desks, are covered in bacteria, and those bacteria are mostly good, but are also some of the bad stuff. So maybe it's a good thing if you have a friend who like sneezes on their hand and then like wants to shake your hand or something or give you a high five. Yeah, that's a good time to use this stuff because you can be pretty certain that whatever's on their hand is not something that you want in your body. But for the most part, it's kind of silly. It's kind of silly to have around, right? Because the majority of the time, bacteria are not bad; they're good. Right? So let's talk about in those instances where they are bad, why they're bad. Okay? There's two things that can make you sick from bacteria. The first one is called exotoxins. Okay? Exotoxins are chemicals that are excreted by the bacteria that make you sick. Chemicals secreted or excreted. Secreted is probably a little better term. It means that it wasn't a waste product. Chemicals secreted by bacteria that make you sick. Now, why would bacteria want to make you sick? Evolutionarily speaking. Yeah. Sure, but how? Yeah, but how does making you sick help them? Yeah.
To distract the white blood cells.
No, it actually activates the white blood cells. There are bacteria that live in you all the time that don't make you sick, and your immune system doesn't really bother them. So why would it be good for bacteria to activate your immune system?
To kill off the competing bacteria.
Yeah. So the answer to that is that it's not. It's not good for them to make you sick, right? In fact, they really don't want to. But some of them do by accident because they secrete these exotoxins in order to compete. We talked about the fact that if I had a bunch of bacteria on my hand and I got staph on my hand, the staph has to compete with the bacteria that's already there. Well, how does it compete with it? When those colonies start growing closer and closer together, they secrete these exotoxins that kill off the other colonies. And by killing off the other colonies, that allows them for their colony to get bigger, right? It just happens to be that in 1% of the cases, those bacterial exotoxins that are being released also make me sick. That's not to say that the bacteria that like live in my gut, for instance, it's not to say that they're not releasing exotoxins. They're all competing with each other. There's a big war happening in my intestines right now with prokaryotes that are trying to take over, but it doesn't affect me because the exotoxins that are being released are not exotoxins that my body responds to or that harm my body. Yeah.
So like if there's a different, like the staph, whatever, that feed out like your regular bacteria, like what happens?
Well, if you had a cut, you'd get a staph infection, right? It would start taking over, and the exotoxins that are in the staph would cause your skin to be inflamed and could cause damage to your tissues and.
Will it eventually go back with the worse, or
What do you mean?
could it like become?
I don't know. Sure.
Like,
You mean like could
if your
could
bacteria, like,
could they fight it off on their own? Maybe. The problem is this, that once they take hold and there's enough of them, the colony is large enough, it's hard for anything to really take over and stop that colony. But if there's just like a couple of them, then it's really easy for them to just be outcompeted. Yeah. All right. So those are exotoxins, right? All bacteria have those. Then there's this other thing that's called endotoxins. Endotoxins are chemicals, specifically lipopolysaccharides, that are released when gram-negative bacteria die. That cause sickness. Okay, so if you kill off gram-negative bacteria too quick, you're in for a world of hurt, right? You can go into sepsis, where essentially you have all of these toxins that are being released by the dead bacteria, where maybe when they were alive, the exotoxins that they were releasing weren't such a big deal, but once they die, they release all these endotoxins, and that makes you much, much sicker, right? So it's very important when things like antibiotics are given that you make sure that you are not going to target gram-negative bacteria, because if you were to kill off gram-negative bacteria too quickly, that could cause symptoms that were far worse than the symptoms that you were experiencing from being sick in the first place, right? So on that, oh, I guess one other thing we'll talk about first. 0% of archaea cause disease. None. There are no pathogenic archaea. All right, so let's talk about antibiotics. There are two main pathways that antibiotics take in order to kill off bacteria, right? Number one, they can prevent the formation of peptidoglycan. Okay, peptidoglycan is the main component of the cell wall, right? And if they prevent the formation of the cell wall, what they do is they stop bacteria from being able to resist what's called osmotic pressure, okay? So let's say we had a balloon, right? I had a balloon, and I just take that balloon and I blow it up, and I keep blowing it up. Eventually, I can blow it up enough that the balloon pops, right? But let's say I have like a steel shoebox, right? Something that's the size of a shoebox, but it's made out of steel, and it's got a hole in one side, and I put the balloon in there, and I blow it up inside the steel box. Am I going to be able to ever blow up that balloon enough to the point where it breaks? No, because it's going to be limited by whatever the interior volume of the box is, right? That's how a cell wall works. So a cell wall is this rigid structure that the plasma membrane continues to get bigger and bigger and bigger based on water coming in through osmosis, but the cell wall limits that, right, and prevents it from getting too big and popping. Okay? If you prevent the formation of peptidoglycan, you weaken the cell wall significantly, and by weakening the cell wall significantly, the osmotic pressure continues to come in and makes that bacteria get bigger and bigger and bigger until it pops, right? And they explode and they die, right? So that is a really effective way of killing bacteria, because without a cell wall, osmotic pressure just destroys them, okay? Preventing the formation of peptidoglycan, which type of bacteria do you think that's most effective against? Gram-positive, because they have high concentrations of peptidoglycan. It really doesn't affect gram-negative bacteria all that much, which is good, because that means we can give people antibiotics that work through this mechanism and not be worried about the release of endotoxins, because endotoxins are only released by gram-negative bacteria, and this type of antibiotic is really only going to target gram-positive bacteria, right? What's up?
Is there any antibiotic that targets the other one?
Yes. So the other way that that antibiotics normally work is through preventing the protein synthesis in bacteria. So they can prevent bacterial protein synthesis. Okay, and if you're preventing bacterial protein synthesis, that seems pretty bad, because, like, wouldn't that prevent your protein synthesis as well? But what organelle does protein synthesis? Ribosomes. And what do we know about bacterial ribosomes? They're different, right? They're unique. So how do these things work? A couple different ways. This one that prevents the formation of peptidoglycan, it blocks the enzyme. It blocks the active site of the enzyme that creates peptidoglycan. And these ones here can block the A site of the ribosome. They can block that spot where the amino acids travel into the ribosome, and they can prevent protein synthesis in that way. Okay, there is antibiotic resistance, right? So bacteria are becoming resistant to these antibiotics, right? And the more we use them, the more resistant bacteria are becoming. And the reason why they're becoming resistant is because or not necessarily the reason, but the mechanism by which they're becoming resistant is by having slight changes in like the structure of the enzyme that stops those antibiotics from blocking the active site, or slight changes in the structure of the ribosome that stops that antibiotic from blocking it, right? And then the important thing to understand is that because bacteria have those things that we refer to as plasmids, that are the small rings of DNA that can get shared in between bacteria, that once one bacteria becomes resistant, it's very likely that that can be shared between bacteria through the plasmid method, through what's called bacterial transformation, right? That we'll talk about in depth a little bit further, a little bit later on. The more we use an antibiotic, the more likely it is for a bacteria to become resistant to it. And once bacteria become resistant to it, it over time will just spread between different species of bacteria. For instance, if you've ever bought soap that says antibacterial on it, it's very likely that that soap does nothing, because that same antibiotic has been in every soap since like the 1960s, right? So every time somebody washes their hands, they are using that antibiotic, the same antibiotic, and therefore almost all bacteria are immune to that specific antibiotic, right? They're all resistant to it, and so it doesn't really do anything anymore. And the more we use an antibiotic, the more likely it is for bacteria to become resistant. So there are some antibiotics, for instance, that are used in livestock. They're given to cows and chickens and stuff like that, and those antibiotics are not really useful in the medical field anymore because they're used so often and in so many cases where they don't need to be in livestock, that there's just so many antibiotic resistant bacteria out there to them that they're not really effective anymore. There are certain antibiotics that we sort of keep in our back pocket, right, that we don't use unless the situation is extremely dire and we've tried out the rest of the antibiotics that we have, right? And those antibiotics are like our last resort. Like, okay, we only use these, you know, whenever nothing else works, because we don't want things to become resistant to them, because it's our secret weapon, right? And so people ask, like, okay, why do antibiotics need to be prescribed? Well, they need to be prescribed because they're not necessarily any more dangerous than like Advil or Tylenol. Like if you take a bunch of Tylenol, you're going to die. If you take a bunch of antibiotics, you're probably going to die, right? So it's not massively more dangerous. But the more you use them, and the more you use them when you don't need to use them, the more likely it is for bacteria to become resistant to them, and then we can't use them anymore, and we have to find new antibiotics. Question is always asked as well is, where do these antibiotics come from? A lot of times antibiotics come from bacteria. They're exotoxins that certain bacteria release in order to compete with other bacteria, right? Or in the case of the most famous antibiotic, which is penicillin, right? That comes from mold, and it's mold has the ability to compete with bacterial colonies and remove bacterial colonies then. Yeah?
So when we wash our hands with regular soap, does that kill bacteria?
It does, yeah. So let's talk about that. Washing your hands with soap or using hand sanitizer, that's called an antiseptic. What's up?
So with like when you say that antibiotics are possible 0% cause diseases, I know that like a lot of people can be allergic to penicillin. Is that different than a disease?
Sure, yeah, it's an allergy. So
Okay.
so you have
So that doesn't count as a disease.
no. It's essentially a false antigen. Right? Just like you wouldn't say dust causes disease, but there's lots of people who are allergic to dust, right? Yeah. What's up?
Since antibiotics use bacterial exotoxins, are we kind of giving other bacteria like a home competition by removing exotoxins?
Yeah, whatever bacteria. I mean, like a lot of times they're pretty rare bacteria. They're not ones that would normally be found, you know, on the surface of a table or on your skin or something like that. But yeah, we are giving some bacteria the leg up, or screwing them over. If that's their exotoxin and then everything becomes resistant to their exotoxin, then that's bad, right? All right, so let's talk about the difference between antibiotics and antiseptics. There was this this thing going around a few years ago where everybody was like, oh, don't use hand sanitizer because you're going to create a super germ, right? Anybody ever hear that from anybody? Like, don't use it because there's certain ones that when it's 99.99 and that 0.01 that lives, like they're going to be immune to the hand sanitizer, and then we're going to create a super germ, right? That's not true, and I'll tell you why. Because this is an antiseptic. It's not an antibiotic. Antiseptics work by destroying plasma membranes and denaturing proteins, right? So these are chemicals like soap and alcohol that denature proteins and dissolve plasma membranes. So this is a chemistry thing, okay? So the plasma membrane, we know, has a phosphate head that's hydrophilic, and it has fatty acid tails that are hydrophobic, right? And the reason why it's so good at separating things from the inside and outside of the cell is because those hydrophobic tails will stay away from water, and the hydrophilic heads will stay away from oil, right? So the only thing that could really mess them up is if there was some molecule out there that was both polar and non-polar, and then that would really mess things up, and it would mess up the plasma membrane and dissolve it. Well, guess what? Alcohol is a molecule that has a polar end and a non-polar end, right? And the polar end of it is really good at interacting with the phosphate heads, and the non-polar end is really good at interacting with the fatty acid tails. And so you can dissolve a plasma membrane within alcohol by just mixing it with it, because alcohol is both polar and non-polar, right? So, is it possible that some bacteria is going to be born that is immune to dissolving in alcohol? I guess maybe, but it's no less likely, or no more likely rather, than like you being born with the ability to jump into hot lava and be fine. Like, chemically speaking, that doesn't make any sense, that your proteins wouldn't denature when you jumped into lava, and it doesn't make any sense that plasma membrane wouldn't dissolve when it's put into alcohol or when soap is used on it. Soap is also really good at breaking up nonpolar and polar compounds. What's up?
So like when you drink alcohol, pretty much dissolve your inside.
No, I mean, yes. If you were to, like, let's say you were to drink like pure alcohol, like, you know, 195 proof or something like that, it's going to do some damage on the way down, right? But like if it's in solution, not really. Your stomach lining's pretty tough. What's up?
So with like those like soap and alcohol, does it mess up your actual skin?
Sure, yeah.
Like,
Try it out. Like, use hand sanitizer twice a period for a few days, and your hands will be destroyed. They'll be all cracky and dry and nasty, right? And why is that? It's because it's destroying the skin cells on your hand, because it destroys cells. That's just what it does. It's alcohol, right? What's up?
So what makes soap better than hand sanitizer?
It's nothing. Soap also removes dirt. So like, so when you wash your hands, right, you put soap on them and then you wash it away, and so you like get all the grime and grit off as well. But when you do this, right, you just like, it's not working, but whatever, right? So you just do this, and then it evaporates away, and whatever dirt was on your hand is still on your hand. So it's not like any cleaner, it's just more sterile. So like that's, I guess, why soap is better than hand sanitizer, but it's, I mean, not really. It depends why you're doing it. If you want to be clean or you want to be sterile. All right. How much time we got left? 1 minute. Okay, we'll stop there.